Biocybernetics. Biocybernetics is the application of cybernetics to biological science disciplines such as neurology and multicellular systems. Biocybernetics plays a major role in systems biology, seeking to integrate different levels of information to understand how biological systems function. The field of cybernetics itself has origins in biological disciplines such as neurophysiology. Biocybernetics is an abstract science and is a fundamental part of theoretical biology, based upon the principles of systemics. Biocybernetics is a psychological study that aims to understand how the human body functions as a biological system and performs complex mental functions like thought processing, motion, and maintaining homeostasis. PsychologyDictionary.org. Within this field, many distinct qualities allow for different distinctions within the cybernetic groups such as humans and insects such as beehives and ants. Humans work together but they also have individual thoughts that allow them to act on their own, while worker bees follow the commands of the queen bee. Seeley, 1989. Although humans often work together, they can also separate from the group and think for themselves. Gackenbach, J. 2007. A unique example of this within the human sector of biocybernetics would be in society during the colonization period, when Great Britain established their colonies in North America and Australia. Many of the traits and qualities of the mother country were inherited by the colonies, as well as niche qualities that were unique to them based on their areas like language and personality, similar vines and grasses, where the parent plant produces offshoots, spreading from the core. Once the shoots grow their roots and get separated from the mother plant, they will survive independently and be considered their plant. Society is more closely related to plants than to animals since, like plants, there is no distinct separation between parent and offspring. The branching of society is more similar to plant reproduction than to animal reproduction. Humans are a K-selected species that typically have fewer offspring that they nurture for longer periods than R-selected species. It could be argued that when Britain created colonies in regions like North America and Australia, these colonies, once they became independent, should be seen as offspring of British society. Like all children, the colonies inherited many characteristics, such as language, customs and technologies, from their parents, but still develop their own personality. This form of reproduction is most similar to the type of vegetative reproduction used by many plants, such as vines and grasses, where the parent plant produces offshoots, spreading ever further from the core. When such a shoot, once it has produced its own roots, gets separated from the mother plant, it will survive independently and define a new plant. Thus, the growth of society is more like that of plants than like that of the higher animals that we are most familiar with. There is not a clear distinction between a parent and its offspring. Superorganisms are also capable of the so-called distributed intelligence, a system composed of individual agents with limited intelligence and information. These can pool resources to complete goals beyond the individual's reach on their own. Similar to the concept of game theory. Derloff, S.N., Bloom, L.E. 2010. In this concept, individuals and organisms make choices based on the behaviors of the other player to deem the most profitable outcome for them as an individual rather than a group. Terminology. Biocybernetics is a conjoined word from bio Greek, bio life, and cybernetics Greek, kybernetic controlling governing. Although the extended form of the word is biological cybernetics, the field is most commonly referred to as biocybernetics in scientific papers. Early proponents. Early proponents of biocybernetics include Ross Ashby, Hans Drischel, and Norbert Wiener among others. Popular papers published by each scientist are listed below. Ross Ashby, Introduction to Cybernetics, 1956 1. Hans Drischel, Einführung in die Biokybernetik, 1972 2. Norbert Wiener, Cybernetics or Control and Communication in the Animal and the Machine, 1948 3. Similar fields. Papers and research that delve into topics involving biocybernetics may be found under a multitude of similar names, including molecular cybernetics, neurocybernetics, and cellular cybernetics. Such fields involve disciplines that specify certain aspects of the study of the living organism. For example, neurocybernetics focuses on the study neurological models in organisms. Categories. Biocybernetics, the study of an entire living organism. Neurocybernetics, cybernetics dealing with neurological models. Psychocybernetics was the title of a self-help book, and is not a scientific discipline. Molecular cybernetics, cybernetics dealing with molecular systems, e.g. molecular biology cybernetics. Cellular cybernetics, cybernetics dealing with cellular systems, e.g. information technology, cell phones or biological cells. Genetic divergence. Genetic divergence is the process in which two or more populations of an ancestral species accumulate independent genetic changes, mutations, through time, often leading to reproductive isolation and continued mutation even after the populations have become reproductively isolated for some period of time, as there is not any genetic exchange anymore. 1. In some cases, subpopulations cover living in ecologically distinct peripheral environments can exhibit genetic divergence from the remainder of a population, especially where the range of a population is very large, see parapatric speciation. The genetic differences among divergent populations can involve silent mutations that have no effect on the phenotype or give rise to significant morphological and/or physiological changes. Genetic divergence will always accompany reproductive isolation, either due to novel adaptations via selection and/or due to genetic drift, and is the principal mechanism underlying speciation. On a molecular genetics level, genetic divergence is due to changes in a small number of genes in a species, resulting in speciation. 2. However, researchers argue that it is unlikely that divergence is a result of a significant, single, dominant mutation in a genetic locus because if that were so, the individual with that mutation would have zero fitness. 3. Consequently, they could not reproduce and pass the mutation on to further generations. Hence, it is more likely that divergence, and subsequently reproductive isolation, are the outcomes of multiple small mutations over evolutionary time accumulating in a population isolated from gene flow. 2. Causes. Founder effect. One possible cause of genetic divergence is the founder effect, which is when a few individuals become isolated from their original population. Those individuals might overrepresent a certain genetic pattern, which means that certain biological characteristics are overrepresented. These individuals can form a new population with different gene pools from the original population. For example, 10% of the original population has blue eyes and 90% has brown eyes. By chance, 10 individuals are separated from the original population. If this small group has 80% blue eyes and 20% brown eyes, then their offspring would be more likely to have the allele for the blue eyes. As a result, the percentage of the population with blue eyes would be higher than the population with brown eyes, which is different from the original population. Bottleneck effect. Another possible cause of genetic divergence is the bottleneck effect. The bottleneck effect is when an event, such as a natural disaster, causes a large portion of the population to die. By chance, certain genetic patterns will be overrepresented in the remaining population, which is similar to what happens with the founder effect. 4. Disruptive selection. A graph Genetic divergence. Genetic divergence is the process in which two or more populations of an ancestral species accumulate independent genetic changes, mutations, through time, often leading to reproductive isolation and continued mutation even after the populations have become reproductively isolated for some period of time, as there is not any genetic exchange anymore. 1. In some cases, subpopulations cover living in ecologically distinct peripheral environments can exhibit genetic divergence from the remainder of a population, especially where the range of a population is very large, see parapatric speciation. The genetic differences among divergent populations can involve silent mutations that have no effect on the phenotype or give rise to significant The genetic differences among divergent populations can involve silent mutations that have no effect on the phenotype or give rise to significant morphological and/or physiological changes. Genetic divergence will always accompany reproductive isolation, either due to novel adaptations via selection and/or due to genetic drift, and is the principal mechanism underlying speciation. On a molecular genetics level, genetic divergence is due to changes in a small number of genes in a species, resulting in speciation. 2. However, researchers argue that it is unlikely that divergence is a result of a significant, single, dominant mutation in a genetic locus because if that were so, the individual with that mutation would have zero fitness. 3. Consequently, they could not reproduce and pass the mutation on to further generations. Hence, it is more likely that divergence, and subsequently reproductive isolation, are the outcomes of multiple small mutations over evolutionary time accumulating in a population isolated from gene flow. 2. Causes. Founder effect. One possible cause of genetic divergence is the founder effect, which is when a few individuals become isolated from their original population. Those individuals might overrepresent a certain genetic pattern, which means that certain biological characteristics are overrepresented. These individuals can form a new population with different gene pools from the original population. For example, 10% of the original population has blue eyes and 90% has brown eyes. By chance, 10 individuals are separated from the original population. If this small group has 80% blue eyes and 20% brown eyes, then their offspring would be more likely to have the allele for the blue eyes. As a result, the percentage of the population with blue eyes would be higher than the population with brown eyes, which is different from the original population. Bottleneck effect. Another possible cause of genetic divergence is the bottleneck effect. The bottleneck effect is when an event, such as a natural disaster, causes a large portion of the population to die. By chance, certain genetic patterns will be overrepresented in the remaining population, which is similar to what happens with the founder effect. 4. Disruptive selection. A graph showing selection for the extremes and against the mean. Genetic divergence can occur without geographic separation, through disruptive selection. This occurs when individuals in a population with both high and low phenotypic extremes are fitter than the intermediate phenotype. 5. These individuals occupy two different niches, within each niche there is Gaussian trait distribution. 6. If the genetic variation between niches is high, then there will be strong reproductive isolation. 6. If genetic variation is below a certain threshold, then introgression will occur, but if variation is above a certain threshold, the population can split, resulting in speciation. 6. Disruptive selection is seen in the bimodal population of Darwin's finches, Geospiza fortis. 7. The two modes specialize in eating different types of seeds, small and soft versus large and hard. This results in beaks of different sizes with different force capacities. 7. Individuals with intermediate beak sizes are selected against. 7. The song structure and response to song also differs between the two modes. 7. There is minimal gene flow between the two modes of G. fortis. 7. References. 1. Survivorship curve. A survivorship curve is a graph showing the number or proportion of individuals surviving to each age for a given species or group, e.g. males or females. Survivorship curves can be constructed for a given cohort, a group of individuals of roughly the same age based on a life table. An illustration of the three idealized types of curve logarithmic scale. There are three generalized types of survivorship curves. 1. Type 1 or convex curves are characterized by high age-specific survival probability in early and middle life, followed by a rapid decline in survival in later life. They are typical of species that produce few offspring but care for them well, including humans and many other large mammals such as elephants. These are also known as K-selected species. C. R. K-selection theory. Type 2 or diagonal curves are an intermediate between types I and III, where a roughly constant mortality rate, survival probability is experienced regardless of age. Some birds and some lizards follow this pattern. Type 3 or concave curves have the greatest mortality, lowest age-specific survival, early in life, with relatively low rates of death, high probability of survival for those surviving this bottleneck. This type of curve is characteristic of species that produce a large number of offspring, known as R-selected species. This includes most marine invertebrates. For example, oysters produce millions of eggs, but most larvae die from predation or other causes. Those that survive long enough to produce a hard shell live relatively long. The number or proportion of organisms surviving to any age is plotted on the y-axis, generally with a logarithmic scale starting with 1,000 individuals, while their age, often as a proportion of maximum lifespan, is plotted on the x-axis. In mathematical statistics, the survival function is one specific form of survivorship curve and plays a basic part in survival analysis. There are various reasons that a species exhibits their particular survivorship curve, but one contributor can be environmental factors that decrease survival. For example, an outside element that is non-discriminatory in the ages that it affects of a particular species is likely to yield a type 2 survivorship curve, in which the young and old are equally likely to be affected. On the other hand, an outside element that preferentially reduces the survival of young individuals is likely to yield a type 3 curve. Finally, if an outside element only reduces the survival of organisms later in life, this is likely to yield a type 1 curve. 2. Actuarial Science Life History Theory Population Ecology 1. B. Rees, Jane, 1st of January. Approximate and ultimate causation. Approximate cause is an event which is closest to, or immediately responsible for causing, some observed result. This exists in contrast to a higher level ultimate cause or distal cause, which is usually thought of as a real reason something occurred. Why? Because graph of the capsizing of the Herald of Free Enterprise, click to see in detail. The concept is used in many fields of research and analysis, including data science and ethology. Example, why did the ship sink? Proximate cause, because it was holed beneath the waterline, water entered the hull and the ship became denser than the water which supported it, so it could not stay afloat. Ultimate cause, because the ship hit a rock which tore open the hole in the ship's hull. In most situations, an ultimate cause may itself be a proximate cause in comparison to a further ultimate cause. Hence we can continue the above example as follows. Example, why did the ship hit the rock? Proximate cause, because the ship failed to change course to avoid it. Ultimate cause, because the ship was under autopilot and the autopilot's data was inaccurate. Even stronger, because the shipwrights made mistakes in the ship's construction. Stronger yet, because the scheduling of labor at the shipyard allows for very little rest. In absurdum, because the shipyard's owners have very small profit margins in an ever-shrinking market. In biology, ultimate causation explains traits in terms of evolutionary forces acting on them. Example, female animals often display preferences among male display traits, such as song. An ultimate explanation based on sexual selection states that females who display preferences have more vigorous or more attractive male offspring. Proximate causation explains biological function in terms of immediate physiological or environmental factors. Example, a female animal chooses to mate with a particular male during a mate choice trial. A possible proximate explanation states that one male produced a more intense signal, leading to elevated hormone levels in the female producing copulatory behavior. Although the behavior in these two examples is the same, the explanations are based on different sets of factors incorporating evolutionary versus physiological factors. These can be further divided. For example, proximate causes may be given in terms of local muscle movements or in terms of developmental biology. See Tinbergen's 4 questions. In philosophy. In analytic philosophy, notions of cause adequacy are employed in the causal model. In order to explain the genuine cause of an effect, one would have to satisfy adequacy conditions, which include, among others, the ability to distinguish between 1. Genuine causal relationships and accidents. 2. Causes and effects. 3. Causes and effects from a common cause. One famous example of the importance of this is the Duhem-Quine thesis, which demonstrates that it is impossible to test a hypothesis in isolation, because an empirical test of the hypothesis requires one or more background assumptions. One way to solve this issue is to employ contrastive explanations. Several philosophers of science, such as Lipton, argue that contrastive explanations are able to detect genuine causes. An example of a contrastive explanation is a cohort study that includes a control group, where one can determine the cause from observing two otherwise identical samples. This view also circumvents the problem of infinite regression of why questions that proximate causes create. Habituation. Habituation is a form of non-associative learning in which an organism's non-reinforced response to a stimulus decreases after repeated or prolonged presentations of that stimulus. For example, organisms may habituate to repeated sudden loud noises when they learn that these have no consequences. 2. Habituation can occur in responses that habituate include those that involve an entire organism or specific biological component systems of an organism. The broad ubiquity of habituation across all forms of life has led to it being called the simplest, most universal form of learning, as fundamental a characteristic of life as DNA. Habituation is a form of non-associative learning in which an organism's non-reinforced response to a stimulus decreases after repeated or prolonged presentations of that stimulus. For example, organisms may habituate to repeated sudden loud noises when they learn that these have no consequences. 2. Habituation can occur in responses that habituate include those that involve an entire organism or specific biological component systems of an organism. The broad ubiquity of habituation across all forms of life has led to it being called the simplest, most universal form of learning, as fundamental a characteristic of life as DNA. 3. Functionally, habituation is thought to free up cognitive resources for other stimuli that are associated with biologically important events by diminishing the response to inconsequential stimuli. A progressive decline of a behavior in a habituation procedure may also reflect nonspecific effects such as fatigue, which must be ruled out when the interest is in habituation. 4. Habituation is relevant in psychiatry and psychopathology, as several neuropsychiatric conditions including autism, schizophrenia, migraine, and Tourette syndrome show reduced habituation to a variety of stimulus types both simple and complex. 5. Drug habituation. There is an additional connotation to the term habituation which applies to psychological dependency on drugs, and is included in several online dictionaries. 6. A team of specialists from the World Health Organization assembled in 1957 to address the problem of drug addiction and adopted the term drug habituation to distinguish some drug use behaviors from drug addiction. According to the WHO lexicon of alcohol and drug terms, habituation is defined as becoming accustomed to any behavior or condition, including psychoactive substance use. 7. By 1964, the report of the Surgeon General of the United States on Smoking and Health 8 included 4 features that characterize drug habituation according to WHO. 1. A desire, but not a compulsion, to continue taking the drug for the sense of improved well-being which it engenders. 2. Little or no tendency to increase the dose. 3. Some degree of psychic dependence on the effect of the drug, but absence of physical dependence and hence of an abstinence syndrome. 4. Detrimental effects, if any, primarily on the individual. However, also in 1964, a committee from the World Health Organization once again convened and decided the definitions of drug habituation and drug addiction were insufficient, replacing the two terms with drug dependence. Substance dependence is the preferred term today when describing drug-related disorders. 9. Whereas the use of the term drug habituation has declined substantially. This is not to be confused with true habituation to drugs, wherein repeated doses have an increasingly diminished effect, as is often seen in addicts or persons taking painkillers frequently. 10. Characteristics. Habituation is a form of non-associative Habituation as a form of non-associative learning can be distinguished from other behavioral changes, e.g., sensory, neural adaptation, fatigue, by considering the characteristics of habituation that have been identified over several decades of research. The characteristics first described by Thompson and Spencer 11 were updated in 2008 and 2009 to include the following. 12. Repeated presentation of a stimulus will cause a decrease in reaction to the stimulus. Habituation is also proclaimed to be a form of implicit learning, which is commonly the case with continually repeated stimuli. This characteristic is consistent with the definition of habituation as a procedure, but to confirm habituation as a process, additional characteristics must be demonstrated. Also observed is spontaneous recovery. That is, a habituated response to a stimulus recovers, increases in magnitude, when a significant amount of time, hours, days, weeks passes between stimulus presentations. Potentiation of habituation is observed when tests of spontaneous recovery are given repeatedly. In this phenomenon, the decrease in responding that follows spontaneous recovery becomes more rapid with each test of spontaneous recovery. Also noted was that an increase in the frequency of stimulus presentation, i.e., shorter interstimulus interval, will increase the rate of habituation. Furthermore, continued exposure to the stimulus after the habituated response has plateaued, i.e., show no further decrement, may have additional effects on subsequent tests of behavior such as delaying spontaneous recovery. The concepts of stimulus generalization and stimulus discrimination will be observed. Habituation to an original stimulus will also occur to other stimuli that are similar to the original stimulus, stimulus generalization. The more similar the new stimulus is to the original stimulus, the greater the habituation that will be observed. When a subject shows habituation to a new stimulus that is similar to the original stimulus but not to a stimulus that is different from the original stimulus, then the subject is showing stimulus discrimination. For example, if one was habituated to the taste of lemon, their responding would increase significantly when presented with the taste of lime. Stimulus discrimination can be used to rule out sensory adaptation and fatigue as an alternative explanation of the habituation process. Another observation mentioned is when a single introduction of a different stimulus late in the habituation procedure when responding to the eliciting stimulus has declined can cause an increase in the habituated response. This increase in responding is temporary and is called dishabituation, and always occurs to the original eliciting stimulus, not to the added stimulus. Researchers also use evidence of dishabituation to rule out sensory adaptation and fatigue as alternative explanations of the habituation process. Habituation of dishabituation can occur. The amount of dishabituation that occurs as a result of the introduction of a different stimulus can decrease after repeated presentation of the dishabituating stimulus. Some habituation procedures appear to result in a habituation process that lasts days or weeks. This is considered long-term habituation. It persists over long durations of time, i.e., shows little or no spontaneous recovery. Long-term habituation can be distinguished from short-term habituation, which is identified by the 9 characteristics listed above. Biological mechanisms. The changes in synaptic transmission that occur during habituation have been well characterized in the Aplysia-Gill and siphon withdrawal reflex. Habituation has been shown in essentially every species of animal and at least, in one species of plants, Mimosa pudica 13, in isolated neuronally differentiated cell lines, as well as in quantum perovskite. 14. The experimental investigation of simple organisms such as the large protozoan Stentor caeruleus provides an understanding of the cellular mechanisms that are involved in the habituation process. 15. Neuroimaging. Within psychology, habituation has been studied through different forms of neuroimaging like PET scan and fMRI. Within fMRI, the response that habituates is the blood oxygen level dependent bold signals triggered by stimuli. Decreases of the bold signal are interpreted as habituation. 16. The amygdala is one of the most studied areas of the brain in relation to habituation. A common approach is to observe the visual processing of facial expressions. A study by Breeder and colleagues used fMRI scans to identify which areas of the brain habituate and at what rate. 17. The results showed that the human amygdala responds and rapidly habituates preferentially to fearful facial expressions over neutral ones. They also observed significant amygdala signal changes in response to happy faces over neutral faces. Blackbird, Allen, Cowan, and Avery 2012 compared the effect of an extremely inhibited temperament and an extremely uninhibited temperament on habituation. Their study found that over repeated presentations individuals with an uninhibited temperament demonstrated habituation in both the amygdala and hippocampus, whereas participants with an inhibited temperament demonstrated habituation in neither brain region. The researchers suggest that this failure to habituate reflects a social learning deficit in individuals with an extremely inhibited temperament, which is a possible mechanism for a higher risk of social anxiety. 18. Debate about learning status. Although habituation has been regarded as a learning process by some as early as 1887, 19, its learning status remained controversial up until the 1920s-1930s. 20. While conceding that reflexes may relax or otherwise decrease with repeated stimulation, the invariance doctrine stipulated that reflexes should not remain constant and that variable reflexes were a pathological manifestation. Indeed, air pilots who showed habituation of post-rotational nystagmus reflex were sometimes ejected from or not recruited for service for World War I, on the grounds that a variable reflex response indicated either a defective vestibular apparatus or a lack of vigilance. 21, 22, 20. Eventually, however, more research from the medical and scientific communities concluded that stimulus-dependent variability reflexes is clinically normal. 23. The opposition to the considering habituation a form of learning was also based on the assumption that learning processes must produce novel behavioral responses and must occur in the cerebral cortex. 21. Non-associative forms of learning such as habituation and sensitization do not produce novel conditioned responses but rather diminish a pre-existing innate responses and often are shown to depend on peripheral, non-cerebral, synaptic changes in the sensory motor pathway. Most modern learning theorists, however, consider any behavioral change that occurs as a result of experience to be learning, so long as it cannot be accounted for by motor fatigue, sensory adaptation, developmental changes or damage. Criteria for verifying a response decline is learning. Importantly, systematic response declines can be produced by non-learning factors such as sensory adaptation, obstruction of stimulus detection, motor fatigue, or damage. 3 diagnostic criteria are used to distinguish response declines produced by these non-learning factors and response declines produced by habituation learning processes. These are 1. Recovery by dishabituation. 2. Sensitivity of spontaneous recovery to rate of stimulation. 3. Stimulus specificity. Early studies relied on the demonstration of 1. Recovery by dishabituation, the brief recovery of the response to the eliciting stimulus when another stimulus is added, to distinguish habituation from sensory adaptation and fatigue. More recently, 2. Sensitivity of spontaneous recovery to rate of stimulation and 3. Stimulus specificity have been used as experimental evidence for the habituation process. 12. Spontaneous recovery is sensitive to spontaneous recovery, showing recovery that is inversely correlated with the amount of response decline. This is the opposite of what would be expected if sensory adaptation or motor fatigue were the cause of the response decline. Sensory adaptation, or neural adaptation, occurs when an organism can no longer detect the stimulus as efficiently as when first presented, and motor fatigue occurs when an organism is able to detect the stimulus but can no longer respond efficiently. Stimulus specificity stipulates that the response decline is not general, due to motor fatigue, but occurs only to the original stimulus that was repeated. If a response decline shows 1. Dishabituation, 2. Spontaneous recovery that is inversely correlated with the extent of decline, and or 3. Stimulus specificity, then habituation learning is supported. Despite the ubiquity of habituation and its modern acceptance as a genuine form of learning, it has not enjoyed the same focus within research as other forms of learning. On this topic, the animal psychologist James McConnell said, "Nobody cares much about habituation." 24. It has been suggested that the apathy held towards habituation is due to 1. Resistance from traditional learning theorists maintain memory requires reproduction of propositional, linguistic content. 2. Resistance from behaviorists who maintain that, true, learning requires the development of a novel response, whereas habituation is a decrease in a pre-existing response. 3. The behavioral measure of habituation, i.e., a response decline, is very susceptible to confound by non-learning factors, e.g. fatigue, which, therefore, make it more difficult to study. Theories. Various models have been proposed to account for habituation, including the stimulus model comparator theory formulated by Yevgeny Sokolov, 25, the Groves and Thompson dual process theory, 26, and the SOP, standard operating procedures, sometimes opponent process model formulated by Alan Wagner, 27. Stimulus model comparator theory. The stimulus model comparator theory emerged from the research of Sokolov, who used the orienting response as the cornerstone of his studies, and operationally defining the orienting response as EEG activity. Orienting responses are heightened sensitivity experienced by an organism when exposed to a new or changing stimulus. Orienting responses can result in overt, observable behaviors as well as psychophysiological responses such as EEG activity and undergo habituation with repeated presentation of the eliciting stimulus. The Sokolov model 25 assumes that when a stimulus is experienced several times, the nervous system creates a model of the expected stimulus, a stimulus model. With additional presentations of the stimulus, the experienced stimulus is compared with the stimulus model. If the experienced stimulus matches the stimulus model, responding is inhibited. At first, the stimulus model is not a very good representation of the presented stimulus, and thus responding continues because of this mismatch. With additional presentations, the stimulus model is improved, there is no longer a mismatch, and responding is inhibited, causing habituation. However, if the stimulus is changed so that it no longer matches the stimulus model, the orienting response is no longer inhibited. Sokolov locates the stimulus model in the cerebral cortex. Dual process theory. The Groves and Thompson dual process theory of habituation posits that 2 separate processes exist in the central nervous system that interacts to produce habituation. The 2 distinct processes are a habituation process and a sensitization process. The dual process theory argues that all noticeable stimuli will elicit both of these processes and that the behavioral output will reflect a summation of both processes. The habituation process is decremental, whereas the sensitization process is incremental, enhancing the tendency to respond. Thus, when the habituation process exceeds the sensitization process, behavior shows habituation, but if the sensitization process exceeds the habituation process, behavior shows sensitization. Groves and Thompson hypothesize the existence of 2 neural pathways, an SR pathway, involved with the habituation process, and a state pathway, involved with sensitization. The state system is seen as equivalent to a general state of arousal. 26. Examples of the habituation process in animals and humans. Habituation has been observed in an enormously wide range of species from motile single-celled organisms such as the amoeba 28 and Stentor 15 to sea slugs 29 to humans. 30. Habituation processes are adaptive, allowing animals to adjust their innate behaviors to changes in their natural world. A natural animal instinct, for example, is to protect themselves and their territory from any danger and potential predators. An animal needs to respond quickly to the sudden appearance of a predator. What may be less obvious is the importance of defensive responses to the sudden appearance of any new, unfamiliar stimulus, whether it is dangerous or not. An initial defensive response to a new stimulus is important because if an animal fails to respond to a potentially dangerous unknown stimulus, the results could be deadly. Despite this initial, innate defensive response to an unfamiliar stimulus, the response becomes habituated if the stimulus repeatedly occurs but causes no harm. An example of this is the prairie dog habituating to humans. Prairie dogs give alarm calls when they detect a potentially dangerous stimulus. This defensive call occurs when any mammal, snake, or large bird approaches them. However, they habituate to noises, such as human footsteps, that occur repeatedly but result in no harm to them. If prairie dogs never habituate to non-threatening stimuli, they would be constantly sending out alarm calls and wasting their time and energy. 31. However, the habituation process in prairie dogs may depend on several factors including the particular defensive response. In one study that measured several different responses to the repeated presence of humans, the alarm calls of prairie dogs showed habituation whereas the behavior of escaping into their burrows showed sensitization. 32. Another example of the importance of habituation in the animal world is provided by a study with harbor seals. In one study researchers measured the responses of harbor seals to underwater calls of different types of killer whales. 33. The seals showed a strong response when they heard the calls of mammal-eating killer whales. However, they did not respond strongly when hearing familiar calls of the local fish-eating population. The seals, therefore, are capable of habituating to the calls of harmless predators, in this case, harmless killer whales. While some researchers prefer to simply describe the adaptive value of observable habituated behavior, others find it useful to infer psychological processes from the observed behavior change. For example, habituation of aggressive responses in male bullfrogs has been explained as an attentional or learning process that allows animals to form enduring mental representations of the physical properties of a repeated stimulus and to shift their focus of attention away from sources of irrelevant or unimportant stimulation. 34. Habituation of innate defensive behaviors is also adaptive in humans, such as habituation of a startle response to a sudden loud noise. But habituation is much more ubiquitous even in humans. An example of habituation that is an essential element of everyone's life is the changing response to food as it is repeatedly experienced during a meal. When people eat the same food during a meal, they begin to respond less to the food as they become habituated to the motivating properties of the food and decrease their consumption. Eating less during a meal is usually interpreted as reaching satiety or getting full, but experiments suggest that habituation also plays an important role. Many experiments with animals and humans have shown that providing variety in a meal increases the amount that is consumed in a meal, most likely because habituation is stimulus-specific and because variety may introduce dishabituation effects. 35. Food variety also slows the rate of habituation in children and may be an important contributing factor to the recent increases in obesity. 36. Richard Solomon and John Corbett 1974 proposed the opponent process theory, arguing that habituation is also found in emotional responses. This theory proposes that not all emotional reactions to a stimulus change in the same way when the stimulus is presented repeatedly. Some weaken, decrease, while others are strengthened, increase. The overall effect is that an outside stimulus provokes an emotional reaction that increases rapidly until it is at its most intense. Gradually, the emotional state declines to a level lower than normal and eventually returns to neutral. This pattern coincides with two internal processes referred to as the A process and B process. Hence, the opponent process theory predicts that subjects will show no reaction following a stimulus after a repetition of this same stimulus. It is the after reaction that is much larger and prolonged than if an initial reaction to a stimulus occurred. 37. Relevance to neuropsychiatry. Habituation abnormalities have been repeatedly observed in a variety of neuropsychiatric conditions including autism spectrum disorder ASD, Fragile X syndrome, schizophrenia, Parkinson's disease PD, Huntington's disease HD, attention deficit hyperactivity disorder ADHD, Tourette's syndrome TS, and migraine. 5. In human clinical studies, habituation is most often studied using the acoustic startle reflex. Acoustic tones are delivered to participants through headphones and the subsequent eye blink response is recorded directly by observation or by electromyography (EMG). Depending on the disorder, habituation phenomena have been implicated as a cause, symptom, or therapy. 5. Reduced habituation is the most common habituation phenotype reported across neuropsychiatric disorders, although enhanced habituation has been observed in HD and ADHD. 5. It also appears that abnormal habituation is often predictive of symptom severity in several neuropsychiatric disorders, including ASD, 38 PD, 39, and HD. 41. Moreover, there are instances where treatments that normalize the habituation deficit also improve other associated symptoms. 42. As a therapy, habituation processes have been hypothesized to underlie the efficacy of behavioral therapies, i.e. habit reversal training, exposure therapy for TS and PTSD. 43. Although extinction processes may be operating instead. Uses and challenges of the habituation procedure. Habituation procedures are used by researchers for many reasons. For example, in a study on aggression in female chimpanzees from a group known as the Kasakila chimpanzee community, researchers habituated the chimpanzees by repeatedly exposing them to the presence of human beings. 44. Their efforts to habituate the chimpanzees before the field researchers studied the animal's behavior was necessary in order for them to eventually be able to note the natural behavior of the chimpanzees, instead of simply noting chimpanzee behavior as a response to the presence of the researchers. In another study, Migumba chimpanzees in the Gombe National Park were habituated for at least 4 years before the introduction of systematic data collection. 45. Researchers also use habituation and dishabituation procedures in the laboratory to study the perceptual and cognitive capabilities of human infants. The presentation of a visual stimulus to an infant elicits looking behavior that habituates with repeated presentations of the stimulus. When changes to the habituated stimulus are made or a new stimulus is introduced, the looking behavior returns dishabituates. A recent fMRI study revealed that the presentation of a dishabituating stimulus has an observable, physical effect upon the brain. 46. In one study, the mental spatial representations of infants were assessed using the phenomenon of dishabituation. 47. Infants were presented repeatedly with an object in the same position on a table. Once the infants habituated to the object, i.e., spent less time looking at it, either the object was spatially moved while the infant remained at the same place near the table, or the object was left in the same place but the infant was moved to the opposite side of the table. In both cases, the spatial relationship between the object and the infant had changed, but only in the former case did the object itself move. Would the infants know the difference? Or would they treat both cases as if the object itself moved? The results revealed a return of looking behavior, dishabituation, when the object's position was changed, but not when the infant's position was changed. Dishabituation indicates that infants perceived a significant change in the stimulus. Therefore, the infants understood when the object itself moved and when it did not. Only when the object itself moved were they interested in it again, dishabituation. When the object remained in the same position as before, it was perceived as the same old boring thing, habituation. In general, habituation-dishabituation procedures help researchers determine the way infants perceive their environments. Habituation is a useful primary tool for then assessing mental processes in the stages of infancy. The purpose for these tests, or paradigms, records looking time, which is the baseline measurement. Habituation of looking time helps to assess certain child capabilities such as memory, sensitivity, and helps the baby recognize certain abstract properties. Habituation is also found to be influenced by unchangeable factors such as infant age, gender, and complexity of the stimulus. Karen and Karen, 1969, Cohen, Deloach, and Rissman, 1975, Friedman, Nagy, and Carpenter, 1970, Miller, 1972, Weatherford and Cohen, 1973. Though there are various challenges that come with habituation, some infants have preferences for some stimuli based on their static or dynamic properties. Infant dishabituation also is not perceived as a direct measure for mental processes as well. In previous theories of habituation, an infant's dishabituation was thought to represent their own realization of the remembered stimulus of stimuli. For example, if infants would be dishabituated to a certain color item to a new item, it would be noticed that they remembered the color and compared the two colors for differences. Also, psychology portal. Another challenge that comes with habituation is the dichotomy of novelty versus familiar stimuli. If an infant preferred a novel still, this meant the infant observed the new spatial relation of the object, but not the object itself. If an infant preferred familiarity, the infant would notice the pattern of the stimuli, instead of the actual new stimuli. 48. The habituation-dishabituation procedure is also used to discover the resolution of perceptual systems. For instance, by habituating someone to one stimulus, and then observing responses to similar ones, one can detect the smallest degree of difference that is detectable. Adaptive system. Aplysia. Consumer demand tests, animals. Contact hypothesis. Desensitization, psychology. Hedonic adaptation. Konrad Lorenz, Behind the Mirror, A Search for a Natural History of Human Knowledge, on Habituation Preference Tests, Animals. Maximum parsimony, phylogenetics. In phylogenetics and computational phylogenetics, maximum parsimony is an optimality criterion under which a phylogenetic tree that minimizes the total number of character state changes or minimizes the cost of differentially weighted character state changes. Under the maximum parsimony criterion, the optimal tree will minimize the amount of homoplasy, i.e., convergent evolution, parallel evolution, and evolutionary reversals. In other words, under this criterion, the shortest possible tree that explains the data is considered best. Some of the basic ideas behind maximum parsimony were presented by James S. Ferris, 1, in 1970 and Walter M. Fitch in 1971. 2. Maximum parsimony is an intuitive and simple criterion, and it is popular for this reason. However, although it is easy to score a phylogenetic tree by counting the number of character state changes, there is no algorithm to quickly generate the most parsimonious tree. Instead, the most parsimonious tree must be sought in tree space, i.e., amongst all possible trees. For a small number of taxa, i.e., fewer than 9, it is possible to do an exhaustive search, in which every possible tree is scored, and the best one is selected. For 9 to 20 taxa, it will generally be preferable to use branch and bound, which is also guaranteed to return the best tree. For greater numbers of taxa, a heuristic search must be performed. Because the most parsimonious tree is always the shortest possible tree, this means that, in comparison to a hypothetical, true, tree that actually describes the unknown evolutionary history of the organisms under study, the best tree according to the maximum parsimony criterion will often underestimate the actual evolutionary change that could have occurred. In addition, maximum parsimony is not statistically consistent. That is, it is not guaranteed to produce the true tree with high probability, given sufficient data. As demonstrated in 1978 by Joe Felsenstein, 3. Maximum parsimony can be inconsistent under certain conditions, such as long branch attraction. Of course, any phylogenetic algorithm could also be statistically inconsistent if the model it employs to estimate the preferred tree does not accurately match the way that evolution occurred in that clade. This is unknowable. Therefore, while statistical consistency is an interesting theoretical property, it lies outside the realm of testability and is irrelevant to empirical phylogenetic studies. 4. Alternate characterization and rationale. In phylogenetics, parsimony is mostly interpreted as favoring the trees that minimize the amount of evolutionary change required, see for example 2. Alternatively, phylogenetic parsimony can be characterized as favoring the trees that maximize explanatory power by minimizing the number of observed similarities that cannot be explained by inheritance and common descent. 5, 6. Minimization of required evolutionary change on the one hand and maximization of observed similarities that can be explained as homology on the other may result in different preferred trees when some observed features are not applicable in some groups that are included in the tree, and the latter can be seen as the more general approach. 7, 8, 9. While evolution is not an inherently parsimonious process, centuries of scientific experience lend support to the aforementioned principle of parsimony, Occam's razor. Namely, the supposition of a simpler, more parsimonious chain of events is preferable to the supposition of a more complicated, less parsimonious chain of events. Hence, parsimony, sensu lato, is typically sought in inferring phylogenetic trees, and in scientific explanation generally. 10. In detail. Parsimony is part of a class of character-based tree estimation methods which use a matrix of discrete phylogenetic characters and character states to infer one or more optimal phylogenetic trees for a set of taxa, commonly a set of species or reproductively isolated populations of a single species. These methods operate by evaluating candidate phylogenetic trees according to an explicit optimality criterion. The tree with the most favorable score is taken as the best hypothesis of the phylogenetic relationships of the included taxa. Maximum parsimony is used with most kinds of phylogenetic data. Until recently, it was the only widely used character-based tree estimation method used for morphological data. Inferring phylogenies is not a trivial problem. A huge number of possible phylogenetic trees exist for any reasonably sized set of taxa. For example, a mere 10 species gives over 2 million possible unrooted trees. These possibilities must be searched to find a tree that best fits the data according to the optimality criterion. However, the data themselves do not lead to a simple, arithmetic solution to the problem. Ideally, we would expect the distribution of whatever evolutionary characters, such as phenotypic traits or alleles, to directly follow the branching pattern of evolution. Thus we could say that if two organisms possess a shared character, they should be more closely related to each other than to a third organism that lacks this character, provided that character was not present in the last common ancestor of all three, in which case it would be a simpliciomorphy. We would predict that bats and monkeys are more closely related to each other than either is to an elephant, because male bats and monkeys possess external testicles, which elephants lack. However, we cannot say that bats and monkeys are more closely related to one another than they are to whales, though the two have external testicles absent in whales, because we believe that the males in the last common ancestral species of the three had external testicles. However, the phenomena of convergent evolution, parallel evolution, and evolutionary reversals collectively termed homoplasy add an unpleasant wrinkle to the problem of inferring phylogeny. For a number of reasons, two organisms can possess a trait inferred to have not been present in their last common ancestor. If we naively took the presence of this trait as evidence of a relationship, we would infer an incorrect tree. Empirical phylogenetic data may include substantial homoplasy, with different parts of the data suggesting sometimes very different relationships. Methods used to estimate phylogenetic trees are explicitly intended to resolve the conflict within the data by picking the phylogenetic tree that is the best fit to all the data overall, accepting that some data simply will not fit. It is often mistakenly believed that parsimony assumes that convergence is rare. In fact, even convergently derived characters have some value in maximum parsimony-based phylogenetic analyses, and the prevalence of convergence does not systematically affect the outcome of parsimony-based methods. 11. Data that do not fit a tree perfectly are not simply noise. They can contain relevant phylogenetic signal in some parts of a tree, even if they conflict with the tree overall. In the whale example given above, the lack of external testicles in whales is homoplastic. It reflects a return to the condition inferred to have been present in ancient ancestors of mammals, whose testicles were internal. This inferred similarity between whales and ancient mammal ancestors is in conflict with the tree we accept based on the weight of other characters, since it implies that the mammals with external testicles should form a group excluding whales. However, among the whales, the reversal to internal testicles actually correctly associates the various types of whales, including dolphins and porpoises, into the group Cetacea. Still, the determination of the best fitting tree, and thus which data do not fit the tree, is a complex process. Maximum parsimony is one method developed to do this. Character data. The input data used in a maximum parsimony analysis is in the form of characters for a range of taxa. There is no generally agreed upon definition of a phylogenetic character, but operationally a character can be thought of as an attribute, an axis along which taxa are observed to vary. These attributes can be physical, morphological, molecular, genetic, physiological, or behavioral. The only widespread agreement on characters seems to be that variation used for character analysis should reflect heritable variation. Whether it must be directly heritable, or whether indirect inheritance, e.g., learned behaviors, is acceptable, is not entirely resolved. Each character is divided into discrete character states, into which the variations observed are classified. Character states are often formulated as descriptors, describing the condition of the character substrate. For example, the character, eye color, might have the states, blue, and brown. Characters can have two or more states, they can have only one, but these characters lend nothing to a maximum parsimony analysis, and are often excluded. Coding characters for phylogenetic analysis is not an exact science, and there are numerous complicating issues. Typically, taxa are scored with the same state if they are more similar to one another in that particular attribute than each is to taxa scored with a different state. This is not straightforward when character states are not clearly delineated or when they fail to capture all of the possible variation in a character. How would one score the previously mentioned character for a taxon or individual with hazel eyes? Or green? As noted above, character coding is generally based on similarity. Hazel and green eyes might be lumped with blue because they are more similar to that color being light, and the character could be then recoded as eye color, light, dark. Alternatively, there can be multi-state characters, such as eye color, brown, hazel, blue, green. Ambiguities in character state delineation and scoring can be a major source of confusion, dispute, and error in phylogenetic analysis using character data. Note that, in the above example, eyes, present, absent, is also a possible character, which creates issues because eye color is not applicable if eyes are not present. For such situations, unknown is scored, although sometimes X, or the latter usually in sequence data, are used to distinguish cases where a character cannot be scored from a case where the state is simply unknown. Current implementations of maximum parsimony generally treat unknown values in the same manner. The reasons the data are unknown have no particular effect on analysis. Effectively, the program treats a As if it held the state that would involve the fewest extra steps in the tree, see below, although this is not an explicit step in the algorithm. Genetic data are particularly amenable to character-based phylogenetic methods such as maximum parsimony because protein and nucleotide sequences are naturally discrete. A particular position in a nucleotide sequence can be either adenine, cytosine, guanine, or thymine, uracil, or a sequence gap. A position residue in a protein sequence will be one of the basic amino acids or a sequence gap. Thus, character scoring is rarely ambiguous, except in cases where sequencing methods fail to produce a definitive assignment for a particular sequence position. Sequence gaps are sometimes treated as characters, although there is no consensus on how they should be coded. Characters can be treated as unordered or ordered. For a binary two-state character, this makes little difference. For a multi-state character, unordered characters can be thought of as having an equal cost, in terms of number of evolutionary events, to change from any one state to any other. Complementarily, they do not require passing through intermediate states. Ordered characters have a particular sequence in which the states must occur through evolution, such that going between some states requires passing through an intermediate. This can be thought of complementarily as having different costs to pass between different pairs of states. In the eye-color example above, it is possible to leave it unordered, which imposes the same evolutionary cost to go from brown-blue, green hazel, etc. Alternatively, it could be ordered brown hazel green blue. This would normally imply that it would cost 2 evolutionary events to go from brown green, 3 from brown blue, but only 1 from brown hazel. This can also be thought of as requiring eyes to evolve through a hazel stage to get from brown to green, and a green stage to get from hazel to blue, etc. For many characters, it is not obvious if and how they should be ordered. On the contrary, for characters that represent discretization of an underlying continuous variable, like shape, size, and ratio characters, ordering is logical. 12 and simulations have shown that this improves ability to recover correct clades, while decreasing the recovering of erroneous clades. 13, 14, 15. There is a lively debate on the utility and appropriateness of character ordering, but no consensus. Some authorities order characters when there is a clear logical, ontogenetic, or evolutionary transition among the states, for example, legs, short, medium, long. Some accept only some of these criteria. Some run an unordered analysis, and order characters that show a clear order of transition in the resulting tree, which practice might be accused of circular reasoning. Some authorities refuse to order characters at all, suggesting that it biases an analysis to require evolutionary transitions to follow a particular path. It is also possible to apply differential weighting to individual characters. This is usually done relative to a cost of 1. Thus, some characters might be seen as more likely to reflect the true evolutionary relationships among taxa, and thus they might be weighted at a value 2 or more. Changes in these characters would then count as 2 evolutionary steps, rather than 1 when calculating tree scores, see below. There has been much discussion in the past about character weighting. Most authorities now weight all characters equally, although exceptions are common. For example, allele frequency data is sometimes pooled in bins and scored as an ordered character. In these cases, the character itself is often downweighted so that small changes in allele frequencies count less than major changes in other characters. Also, the third codon position in a coding nucleotide sequence is particularly labile, and is sometimes downweighted, or given a weight of 0, on the assumption that it is more likely to exhibit homoplasy. In some cases, repeated analyses are run, with characters reweighted in inverse proportion to the degree of homoplasy discovered in the previous analysis, termed successive weighting. This is another technique that might be considered circular reasoning. Character state changes can also be weighted individually. This is often done for nucleotide sequence data. It has been empirically determined that certain base changes AC, AT, GC, GT, and the reverse changes occur much less often than others AG, CT, and their reverse changes. Growth curve biology. A growth curve is an empirical model of the evolution of a quantity over time. Growth curves are widely used in biology for quantities such as population size or biomass, in population ecology and demography, for population growth analysis, individual body height or biomass, in physiology, for growth analysis of individuals. Values for the measured property. Figure 1. A biphasic bacterial growth curve. Bacterial growth. In this example Figure 1, see lab operon for details. The number of bacteria present in a nutrient-containing broth was measured during the course of an 8-hour cell growth experiment. The observed pattern of bacterial growth is biphasic because 2 different sugars were present, glucose and lactose. The bacteria prefer to consume glucose phase 1 and only use the lactose phase 2 after the glucose has been depleted. Analysis of the molecular basis for this biphasic growth curve led to the discovery of the basic mechanisms that control gene expression. Cancer cell growth. Cancer research is an area of biology where growth curve analysis plays an important role. In many types of cancer, the rate at which tumors shrink following chemotherapy is related to the rate of tumor growth before treatment. Tumors that grow rapidly are generally more sensitive to the toxic effects that conventional anti-cancer drugs have on the cancer cells. Many conventional anti-cancer drugs, for example, 5-fluorouracil, interfere with DNA replication and can cause the death of cells that attempt to replicate their DNA and divide. A rapidly growing tumor will have more actively dividing cells and more cell death upon exposure to such anti-cancer drugs. Figure 2. Tumor growth curve. In the example shown in Figure 2, a tumor is found after the cell growth rate has slowed. Most of the cancer cells are removed by surgery. The remaining cancer cells begin to proliferate rapidly and cancer chemotherapy is started. Many tumor cells are killed by the chemotherapy, but eventually some cancer cells that are resistant to the chemotherapy drug begin to grow rapidly. The chemotherapy is no longer useful and is discontinued. Children who fall significantly below the normal range of growth curves for body height can be tested for growth hormone deficiency and might be treatable with hormone injections. 1. Female stature versus age, USCDC. Male stature versus age, USCDC. Some growth curves for certain biological systems display periods of exponential growth. Typically, periods of exponential growth are of limited duration due to depletion of some rate-limiting resource. 2. The growth of children. Exponential growth. Growth. Evolution of primates. The evolutionary history of the primates can be traced back 57 to 90 million years. One of the oldest known primate-like mammal species, Plesiodapus, came from North America. 2. Another, Archispus, came from China. 3. Other similar basal primates were widespread in Eurasia and Africa during the tropical conditions of the Paleocene and Eocene. Purgatorius is the genus of the 4 extinct species believed to be the earliest example of a primate or a protoprimate, a primatomorph precursor to the Plesiodapiforms, dating to as old as 66 million years ago. Phylogenetic tree of the primates. Notharctus. The surviving tropical population of primates, which is seen most completely in the upper Eocene and lowermost Oligocene fossil beds of the Fayum Depression southwest of Cairo, gave rise to all living species: lemurs of Madagascar, lorises of Southeast Asia, galagos or bush babies of Africa, and the anthropoids, platyrine or New World monkeys, catarrhines or Old World monkeys, and the apes, including Homo sapiens. Origins. The origins and early evolution of primates is shrouded in mystery due to lack of fossil evidence. They are believed to have split from Plesiodapiforms in Eurasia around the early Eocene or earlier. The first true primates so far found in the fossil record are fragmentary and already demonstrate the major split between strepsiorhines and haplorhines. Evolution of strepsiorhines. The earliest strepsiorhines are known as Adapiforms, a diverse group that ranged throughout Eurasia and North America. An early branch of this clade gave rise to lemuriform primates, which includes lemurs and their kin. David Begun has theorized that early primates flourished in Eurasia and that a lineage leading to the African apes and humans, including Dryopithecus, migrated south from Europe or Western Asia into Africa. 4. However, the early European fauna is exemplified by Darwinius, a basal strepsiorhine dated to 47 million years, early Eocene 5. Evolution of haplorhines. The earliest haplorhine primates from the fossil record are the Omimids, which resembled modern-day tarsiers. Like the strepsiorhine Adapiforms, Omimids were diverse and ranged throughout Eurasia and North America. The phylogeny of Omimids, tarsiers, and simians is currently unknown. For many years, it was assumed that primates had first evolved in Africa, and this assumption and the excavations that resulted from it yielded many early simian fossils that chronicled their evolution. Due to the lack of fossils linking simians to the earliest haplorhines, a more recently discovered stem group called Eosimiids found in Asia are thought to have dispersed to Africa and evolved into simians. Eosimiids were very small and similar to tarsiers, though their dentition more closely resembles that of simians. Evolution of color vision. Some of the primates' vertebrate ancestors were tetrachromats, but their nocturnal mammalian ancestors lost 2 of their 4 cones during the Mesozoic. Most modern primates, however, have evolved to be trichromats. All Old World monkeys and apes are trichromats, but New World monkeys are polymorphic trichromats, meaning that males and homozygous females are dichromats while heterozygous females are trichromats, with the exceptions of howler monkeys and night monkeys, who have more and less robust color vision respectively. There are 4 prevailing theories as to what the evolutionary pressure was for primates to develop trichromatic vision. The fruit theory suggests that it was easier for trichromatic primates to find ripe fruit against a green background. While there is data supporting the fruit theory, there is some dispute about whether or not trichromacy was more advantageous for determining how ripe fruit was up close or spotting fruit from afar. The young leaf hypothesis suggests that primates with more advanced color vision could better spot younger and more nutritious leaves during fruit shortages, while there are also theories that suggest more advanced color vision was better for recognizing changes in skin tone, allowing primates to better determine the blood oxygen saturation of others. Still, other theories suppose that primates' color vision evolved alongside their sense of smell, though research has shown no direct correlation between concentration of olfactory receptors and acquisition of color vision. Evolution of New World monkeys. Following the emergence of simiforms in Africa, Platyrini split from Catarrhini during the Eocene when New World monkeys dispersed to South America, likely by rafting on mats of vegetation across the Atlantic Ocean. The Atlantic is estimated to have been possibly 1,000 kilometers, 600 miles narrower, based on estimates from the expansion of the Atlantic mid-ocean ridge formation processes, 25 centimeters, 1 n per year. It is also possible that during this rafting process, there were a number of islands between Africa and South America which have since been submerged. Bayesian estimates of divergence time using conservative but realistic fossil constraints have indicated the most recent common ancestor of New World monkeys to have existed between 27 to 31 million years ago. 6. Evolution of Old World monkeys. The earliest known catarrhine is Cameloyopithecus from uppermost Oligocene at Aragalate in the northern Kenya Rift Valley, dated to 24 million years ago. 7. Its ancestry is thought to be species related to A. Aegyptopithecus, Proplyopithecus, and Parapithecus from the Fayum Depression, at around 35 million years ago. 8. In 2010, Sodonaeus was described as a close relative of the last common ancestor of the crowned catarrhines, and tentatively dated to 29 to 28 million years ago, helping to fill an 11 million year gap in the fossil record. 9. Notable species also include Ensungwepithecus gunnelli and Rukwapithecus flegeli of the Oligocene. 10. Reconstructed tailless proconsul skeleton. In the early Miocene, about 22 million years ago, the many kinds of arboreally adapted primitive catarrhines from East Africa suggest a long history of prior diversification. Fossils dated to be 20 million years old include fragments attributed to Victoriopithecus, believed to be the earliest Old World monkey. Among the genera thought to be in the ape lineage leading up to 13 million years ago are Proconsul, Rangwapithecus, Dendropithecus, Limnopithecus, Nakalapithecus, Equatorius, Nyanzopithecus, Afropithecus, Heliopithecus, and Kenyapithecus, all from East Africa. The presence of other generalized non-cercopithecids of the Middle Miocene age from sites far distant, Odopithecus from cave deposits in Namibia, and Pierolopithecus and Dryopithecus from France, Spain and Austria, is evidence of a wide diversity of forms across Africa and the Mediterranean basin during the relatively warm and equable climatic regimes of the early and Middle Miocene. The youngest of the Miocene hominoids, Oreopithecus, is from coal beds in Italy that have been dated to 9 million years ago. Molecular evidence indicates that the lineage of gibbons, family Hylobatidae, diverged from great apes some 18 to 12 million years ago, and that of orangutans, subfamily Punganate, diverged from the other great apes at about 12 million years. There are no fossils that clearly document the ancestry of gibbons, which may have originated in a so far unknown Southeast Asian hominoid population, but fossil proto-orangutans may be represented by Savapithecus from India and Gripopithecus from Turkey, dated to around 10 million years ago. 11. Human evolution. Human evolution is the evolutionary process that led to the emergence of anatomically modern humans, beginning with the evolutionary history of primates, in particular genus Homo, and leading to the emergence of Homo sapiens as a distinct species of the hominid family, the great apes. This process involved the gradual development of traits such as human bipedalism and language. 12. The study of human evolution involves many scientific disciplines, including physical anthropology, primatology, archaeology, paleontology, neurobiology, ethology, linguistics, evolutionary psychology, embryology and genetics. 13. Genetic studies show that primates diverged from other mammals about 85 million years ago, in the late Cretaceous period, and the earliest fossils appear in the Paleocene, around 55 million years ago. 14. Within the superfamily Hominoidea apes, the family Hominidae diverged from the family Hylobatidae gibbons, some 15 to 20 million years ago. African great apes, subfamily Homininae, diverged from orangutans Punganate, about 14 million years ago. The tribe Homininic humans, Australopithecines and other extinct biped genera, and chimpanzee parted from the tribe Gorillinic gorillas between 9 million years ago and 8 million years ago, and, in turn, the subtribes Hominina humans and biped ancestors and Penina chimpanzees separated about 7.5 million years ago to 5.6 million years ago. 15. Evolution of the pelvis. In primates, the pelvis consists of 4 parts, the left and the right hip bones which meet in the midline ventrally and are fixed to the sacrum dorsally and the coccyx. Each hip bone consists of 3 components, the ilium, the ischium, and the pubis, and at the time of sexual maturity these bones become fused together, though there is never any movement between them. In humans, the ventral joint of the pubic bones is closed. The most striking feature of evolution of the pelvis in primates is the widening and the shortening of the blade called the ilium. Because of the stresses involved in bipedal locomotion, the muscles of the thigh move the thigh forward and backward, providing the power for bipedal and quadrupedal locomotion. 16. See also. Evolution of mammals. List of fossil primates. Evolution. Speciation. In evolutionary biology, sympatric speciation is the evolution of a new species from a surviving ancestral species while both continued to inhabit the same geographic region. In evolutionary biology and biogeography, sympatric and sympatry are terms referring to organisms whose ranges overlap so that they occur together at least in some places. If these organisms are closely related, e.g. sister species, such a distribution may be the result of sympatric speciation. Etymologically, sympatry is derived from Greek syn, son, together, and patris, fatherland. 1. The term was coined by Edward Bagnall Pelton in 1904, who explains the derivation. 1. In sympatric speciation, reproductive isolation evolves within a population without the aid of geographic barriers. Sympatric speciation is one of 3 traditional geographic modes of speciation. 2. 3. Allopatric speciation is the evolution of species caused by the geographic isolation of 2 or more populations of a species. In this case, divergence is facilitated by the absence of gene flow. Parapatric speciation is the evolution of geographically adjacent populations into distinct species. In this case, divergence occurs despite limited interbreeding where the 2 diverging groups come into contact. In sympatric speciation, there is no geographic constraint to interbreeding. These categories are special cases of a continuum from 0 sympatric to complete allopatric spatial segregation of diverging groups. 3. In multicellular eukaryotic organisms, sympatric speciation is a plausible process that is known to occur, but the frequency with which it occurs is not known. 4. In bacteria, however, the analogous process defined as the origin of new bacterial species that occupy definable ecological niches might be more common because bacteria are less constrained by the homogenizing effects of sexual reproduction and are prone to comparatively dramatic and rapid genetic change through horizontal gene transfer. 5. Evidence. Sympatric speciation events are quite common in plants, which are prone to acquiring multiple homologous sets of chromosomes, resulting in polyploidy. The polyploid offspring occupy the same environment as the parent plants, hence symmetry, but are reproductively isolated. A number of models have been proposed for alternative modes of sympatric speciation. The most popular, which invokes the disruptive selection model, was first put forward by John Maynard Smith in 1966. 6. Maynard Smith suggested that homozygous individuals may, under particular environmental conditions, have a greater fitness than those with alleles heterozygous for a certain trait. Under the mechanism of natural selection, therefore, homozygosity would be favored over heterozygosity, eventually leading to speciation. Sympatric divergence could also result from a sexual conflict. 7. Disruption may also occur in multiple gene traits. The medium brown finch Geospiza fortis is showing gene pool divergence in a population on Santa Cruz Island. Beak morphology conforms to 2 different size ideals, while intermediate individuals are selected against. Some characteristics termed magic traits, such as beak morphology, may drive speciation because they also affect mating signals. In this case, different beak phenotypes may result in different bird calls, providing a barrier to exchange between the gene pools. 8. A somewhat analogous system has been reported in horseshoe bats, in which echolocation call frequency appears to be a magic trait. In these bats, the constant frequency component of the call not only determines prey size but may also function in aspects of social communication. Work from one species, the large-eared horseshoe bat Rhinolophus philippinensis, shows that abrupt changes in call frequency among sympatric morphs is correlated with reproductive isolation. 9. A further well-studied circumstance of sympatric speciation is when insects feed on more than one species of host plant. In this case, insects become specialized as they struggle to overcome the various plants' defense mechanisms. Dress and Mallet, 2002, 10. Rhagoletis pomonella, the apple maggot, may be currently undergoing sympatric or, more precisely, heteropatric speciation. The apple feeding race of this species appears to have spontaneously emerged from the hawthorn feeding race in the 1800-1850 AD time frame, after apples were first introduced into North America. The apple feeding race does not now normally feed on hawthorns, and the hawthorn feeding race does not now normally feed on apples. This may be an early step towards the emergence of a new species. 11, 12, 13. Some parasitic ants may have evolved via sympatric speciation. 14. Isolated and relatively homogeneous habitats such as crater lakes and islands are among the best geographical settings in which to demonstrate sympatric speciation. For example, Nicaragua crater lake cichlid fishes include 9 described species and dozens of undescribed species that have evolved by sympatric speciation. 15. 16. Monostroma latissimum, a marine green algae, also shows sympatric speciation in southwest Japanese islands. Although panmictic, the molecular phylogenetics using nuclear introns revealed staggering diversification of population. 17. African cichlids also offer some evidence for sympatric speciation. They show a large amount of diversity in the African Great Lakes. Many studies point to sexual selection as a way of maintaining reproductive isolation. Female choice with regards to male coloration is one of the more studied modes of sexual selection in African cichlids. Female choice is present in cichlids because the female does much of the work in raising the offspring, while the male has little energy input in the offspring. She exerts sensory bias when picking males by choosing those that have colors similar to her or those that are the most colorful. 18, 19, 20. This helps maintain sympatric speciation within the lakes. Cichlids also use acoustic reproductive communication. The male cichlid quivers as a ritualistic display for the female which produces a certain number of pulses and pulse period. Female choice for good genes and sensory bias is one of the deciding factors in this case, selecting for calls that are within her species and that give the best fitness advantage to increase the survivability of the offspring. 21, 22. Male-male competition is a form of intersexual selection and also has an effect on speciation in African cichlids. Ritualistic fighting among males establishes which males are going to be more successful in mating. This is important in sympatric speciation because species with similar males may be competing for the same females. There may be a fitness advantage for one phenotype that could allow one species to invade another. 23, 24. Studies show this effect in species that are genetically similar, have the capability to interbreed, and show phenotypic color variation. Ecological character displacement is another means for sympatric speciation. Within each lake there are different niches that a species could occupy. For example, different diets and depth of the water could help to maintain isolation between species in the same lake. Allochrony offers some empirical evidence that sympatric speciation has taken place, as many examples exist of recently diverged, sister taxa, allochronic species. A case of ongoing sympatric divergence due to allochrony might be found in the marine insect Clunio marinus. 25. A rare example of sympatric speciation in animals is the divergence of resident and transient orca forms in the Northeast Pacific. 26. Resident and transient orcas inhabit the same waters, but avoid each other and do not interbreed. The two forms hunt different prey species and have different diets, vocal behavior, and social structures. Some divergences between species could also result from contrasts in microhabitats. A population bottleneck occurred around 200,000 years ago, greatly reducing the population size at the time as well as the variance of genes which allowed several ecotypes to emerge afterwards. 27. The European polecat Mustela puturius exhibited a rare dark phenotype similar to the European mink Mustela lutriola phenotype, which is directly influenced by peculiarities of forest brooks. 28. Controversy. For some time it was difficult to prove that sympatric speciation was possible, because it was impossible to observe it happening. 3. It was believed by many, and championed by Ernst Mayr, that the theory of evolution by natural selection could not explain how two species could emerge from one if the subspecies were able to interbreed. 29. Since Mayer's heyday in the 1940s and 50s, mechanisms have been proposed that explain how speciation might occur in the face of interbreeding, also known as gene flow. 30. And even more recently concrete examples of sympatric divergence have been empirically studied. 31, 32. The debate now turns to how often sympatric speciation may actually occur in nature and how much of life's diversity it may be responsible for. History. The German evolutionary biologist Ernst Mayr argued in the 1940s that speciation cannot occur without geographic, and thus reproductive, isolation. 29. He stated that gene flow is the inevitable result of symmetry, which is known to squelch genetic differentiation between populations. Thus, a physical barrier must be present, he believed, at least temporarily, in order for a new biological species to arise. 33. This hypothesis is the source of much controversy around the possibility of sympatric speciation. Mayer's hypothesis was popular and consequently quite influential, but is now widely disputed. 34. The first to propose what is now the most pervasive hypothesis on how sympatric speciation may occur was John Maynard Smith, in 1966. He came up with the idea of disruptive selection. He figured that if two ecological niches are occupied by a single species, diverging selection between the two niches could eventually cause reproductive isolation. 35. By adapting to have the highest possible fitness in the distinct niches, two species may emerge from one even if they remain in the same area, and even if they are mating randomly. 30. Defining symmetry. Investigating the possibility of sympatric speciation requires a definition thereof, especially in the 21st century, when mathematical modeling is used to investigate or to predict evolutionary phenomena. 34. Much of the controversy concerning sympatric speciation may lie solely on an argument over what sympatric divergence actually is. The use of different definitions by researchers is a great impediment to empirical progress on the matter. The dichotomy between sympatric and allopatric speciation is no longer accepted by the scientific community. It is more useful to think of a continuum, on which there are limitless levels of geographic and reproductive overlap between species. On one extreme,